Good evening, America, and thank you for joining me tonight on Long-Lived Animals Playing on the Animal Planet. Tonight, we will be discussing the recently acquired animals, the Galapagos tortoises, here at the UNH Zoo. Here, I have with me the curator, Joseph Magrino, and he is the head of the herpetology department, and he will be discussing his various studies, including longevity in the Galapagos tortoise. How are you, Joey?
Doing well, Jason. How are you doing today?
Very well.
Great.
So, congrats.
Thank you. Yeah, we're very excited about to have these Galapagos turtles here. We really want to understand what makes them live so long and see how we can apply it to human longevity.
Okay. So we're going to take a quick commercial break, and we're going to come back and meet the people that take care of these animals day in and day out. Good evening, America. Thank you for joining us on the long-lived episode on Animal Planet. Today I have with us Joey Magrino, the curator of the herpetology department, and today he's going to further discuss, along with his zookeepers, how exactly they're going to use these tortoises in their longevity research. So they want to see how exactly these tortoises are able to live for so long. So Joey, if you could give me a little insight onto what you guys are doing.
Absolutely, Jason. So today I have with me both Rumana and Hibo, two researchers at my in my lab. So Rumana has been looking primarily at the tortoises in regards to their field work, but she's also been doing a lot of work in some of the cellular biology too. And so she's going to be here today explaining a little bit about the tortoises, and then she's going to also give us a kind of a brief definition about aging so people can understand why do we have to do these longevity studies.
Okay.
Great, thank you, Joy. Hello, Jason, and hello, America again. So today I would like to just give a brief overview on these Galapagos tortoises. They originated from the Galapagos Island off the coast of Ecuador, and they are one of the largest living species alive right now. So these species, or the males of the tortoises, they can actually weigh up to 500 pounds, while the females can range anywhere between 250 to 500 pounds, which is way more than us humans. So looking into their lifespan, their lifespan is an average of 100 years and can range all the way up to 150 years. So comparing that to us humans, that's a lot as well, because us humans, our average lifespan is about 79 to 80 years. So now the question is, like, what is aging? Like, how does this have an effect with these? So aging is basically like the slow progression of cells reproducing. So we're looking at the half-life limit and how each species have different go through different reproduction. So humans, for example, we go through like 50 cell cycles. Mouse, for example, go through like 15 to 20 cell cycles. And
So cell cycles, you mean, so if you have a cell dividing, once that cell divides, that cell that has arisen from that division can only further divide 50 times?
correct.
Okay.
And then for tortoises that I'm studying, so far we've seen them to divide about 110 times.
So that same cell can divide that many times?
Yes, that
so
is
that
why.
can contribute to their longevity?
Correct.
Wow, okay.
Yeah, and you know what Ramana has been also seeing, and we've known this fact for a good number of years now, is that the tortoises themselves really don't have a very high caloric intake. They don't eat that much.
Okay.
And in regards to humans, we eat a lot more than we really need to.
Okay.
And what you have to understand in regards to that is for evolution, okay, evolution made a conscious decision. And it said, you know, we can either have longer lived organisms for them to be able to find a mate and reproduce,
Yep.
or we could have these shorter lived species that can build up their muscle mass and have higher brains, and hopefully they'll find their mate faster and reproduce, right? So the split we saw that occurred, and what we're seeing is the tortoises are those longer lived organisms. So they have a slower basal metabolism,
Okay.
and for humans we have a faster basal metabolism. But what happens in that case is we have a higher influx of glucose, or that energy molecule inside of our cells, and that actually at a molecular level attributes to the aging process. And Hibo over here has been really looking into that process in particular. So Hibo, why don't you explain to Jason, you know, what happens at the molecular level in regards to caloric intake?
Well, so far we haven't really studied mammals in research based on like what their caloric intake levels would occur if it were to be lower. So for humans right now, when we have a lower caloric intake, we lead to a lower basal metabolic rate. And once that occurs, we also have a lower glucose levels, and that causes the cells to go through survival mode. Once that occurs, we usually have less reactive oxygen species from occurring, as well as less DNA damage. So it would be really interesting if we could actually look at the Galapagos turtles as a model for studying these mammals, since we haven't really looked into it.
Huh. Yes, let me expand upon that a little bit, and a little bit about her research. So what you have to understand is when we take in glucose, right, it gets converted into what we call these reactive oxygen species. And these are really these high energy molecules that damage our cells and can contribute to the aging process. And she mentioned that they damage their DNA. And so we have very particular ends of our DNA called telomeres. And so you can kind of think of these as like buffers or protection onto your DNA. So what happens is every time your cells divide, they actually get a little bit shorter. Your telomeres shrink just a little bit. Yeah. But if we didn't have them, what you could imagine is the kind of the important stuff inside your genome would eventually shrink. Yeah, exactly. So these act as kind of a buffer zone for them. But when we have high glucose metabolism, they get damaged and they get pre-shortened, which can attribute to the aging factors. Some of the other things that we kind of see with these reactive oxygen species is that they're involved with a process called lipid hydrolysis. So your cells are made out of these lipid membranes. And when reactive oxygen species react with them, they slowly start to disintegrate. And that kind of attributes to a process that we want to call senescence, or the process at which cells stop dividing. And you know, there's multiple ways research is starting to show of what actually attributes to senescence. And there's three mechanisms. One of them being replication senescence, which Rumana touched upon with the Hayflick's limit. But other ones include stress-induced senescence, which means that there are some outside stressors in the extracellular environment that causes the cells to stop dividing, that they're getting signals to stop doing it.
I see.
And then the last one is a protein-secreted senescence, which means that there is other cells actually telling the cells to stop dividing. Now, how does this attribute to aging and human diseases? Well, these senescent cells, as they collect, we're starting to find that those attribute to things like neurodegeneration diseases, cancer, well, not necessarily cancers, I should say, but neurodegenerative diseases. Because ultimately what happens is these cells stop dividing, they collect in a tissue, and what we're finding is that they are secreting out factors that will tell other cells to die.
Okay. Now, quick question before we lead on to the commercial break.
Yeah, absolutely.
So, you say that these telomeres are shrinking,
Yeah.
shortening. Could you just possibly just take in a medicine or enzyme that'll somehow make these bigger? So, like, if you can reconstruct or make these chromosomes larger at all?
So it's
prevent this shortening at all?
That's a really good question. We do have an enzyme in our cells, a protein called telomerase. And telomerase's job is to extend your telomeres, right? That's part of that makes an issue when we look into cloning, right? But at some point, telomerase is told inside the cells to stop working, and it no longer is extending your telomeres. Now, it almost seems like, why don't we just take more of it, and then we can really extend our chromosomes and we could live forever. There's twofold issues with that. One being, it's not the complete issue. You know, there are oxidative stress damages to other
pathways that attribute to the aging.
Yep. And then on top of it, if we continue to take this telomerase enzyme, there's a great disease that has an overactivity of this, cancer. Yeah. So we don't really want to be expressing this at high levels constantly. It's tightly regulated.
Okay. Well, I have some exciting news for you guys. We're going to Skype into a collaborator at the University of Arkansas that's actually looking into lobsters and their longevity. So we'll be back in 5 minutes from the commercial break, and we'll be able to discuss these collaborations. Good evening, America, and thank you for joining us here on Animal Planet, again starring the Long-Lived Animal series. I am here along with my panel members from the University of New Haven Zoo, and again, their research lies on longevity within the tortoises. And here I have Dr. McGinley from the University of Arkansas. Hello. And essentially, he's looking at the same aspect of longevity, however, looking at these aspects within a lobster. So, Dr. McGinley, how's it going?
It's going pretty good, Jay. How are you doing?
I'm doing well. So, give us some background on why exactly a lobster is so efficient in studying longevity.
Absolutely. So lobsters are part of an exclusive group of animals that we call biologically immortal. This means that they only die if they're eaten or injured from a predator or contract a rare disease, but never from old age. They don't have that built-in life expectancy in their cells that humans have. So essentially, they don't die from their own metabolism. Because of this, lobster is able to live and grow for up to 100 years in the wild. But lobsters as old as 130 or 140 years have been caught. So the question remains, why do they grow so large and live so long? And the answer is, it's the telomerase, the enzyme that helps rebuild and maintain telomeres.
Oh, telomerase. So aren't you guys looking at this same exact enzyme within the Galapagos tortoises?
Yeah, absolutely. So we've begun looking at how the Galapagos turtle and telomerase play a role together. But what research have you guys found in respects to lobsters?
Yeah, so basically right now we're looking at the mechanisms in lobsters that control the cell cycle and genes that regulate these mechanisms to determine whether they could play a role in helping people live longer, as well in the same way that they help lobsters. So essentially in humans, telomerase production is turned off when you're an adult. So our telomeres get shorter with time, creating older cells that result in everything we attribute to aging. In lobsters, however, telomerase production is ramped up for their entire lives. So it's a constant, abundant stream that helps keep their telomeres long and stops their cells from ever dying. Interestingly, when lobsters molt their shells, their cells become extremely active. But once the molting process ends, their cells actually enter senescence. So this means that lobsters don't grow much between moltings, so when they're shedding that shell. And the process actually increases the longevity of their cells, as well as prevents cancer due to increased telomerase levels since they are not actively using the enzyme. So even though they have high levels of telomerase, since their cells aren't active between moltings, they're not seeing high levels of cancer like you would in a person.
I see.
Very interesting.
That's very interesting. Huh. All right, awesome. Well, thank you very much, Dr. McGinley, for your input. And that's going to end the series of the long-lived animals here on Animal Planet. And join us next week, and we will be discussing the short-lived animals, which include C. elegans. Thank you very much.